Hello everyone, welcome to the class. Hello Shanta, how are you? Hello YouTube people, welcome to the class. Okay guys, let us start the class. Have you read and come this chapter? Have you read and come this chapter guys? Okay, fine. So let us start the class. In the last class, we have discussed about the inheritance biology, right? Yes, inheritance, fine. So then we can recall and revise this chapter today. Okay. So before going to start the class, can you tell me whether I'm audible and screen is visible, guys? So then we can start the class. Okay. Tell me whether I'm audible and screen is visible. You two people. Fine. Fine. Let us start the class. Okay. So before going to start the class, guys, I wanted to ask you some questions. So that is the experimental basis. Okay. So in search of genetic material, we have some experiment, right? Okay. So now tell me one by one experiment name. Okay, guys? Yes. Actually, this experiment was started in the 1926. Okay. So not exactly. I'm telling that. So this was means the experiment of it was started in the 1926. Okay. So actually, the experiment of the Mendel and the Sutton and Boveri and the T.H. Morgan have narrowed the information of that. So where the DNA is going to be present, it is going to be present in the chromosome. So those all the things we have come to know that, right? It is present in the nucleus. So this all the experiment have not prove that DNA as a genetic material, right? So then though everyone don't know that which is the genetic material, right? So then the experiment was started in the 1928. Okay, guys. So then in search of the genetic material was started in the year 1928. Right, guys? Can you tell me which experiment was the first experiment, guys? Can you tell me which is the first experiment? So that is the transformation principle, right? Transformation principle. Transformation principle. It was given by. Can anyone tell me who is the scientist, guys? Shanta, can you tell me who is the scientist? Griffith, right? Frederick Griffith. Frederick Griffith was started this experiment, right, guys? So now you tell me. Wait. Yeah. So which are the things? Which is the bacteria was chosen for the transformation principle, guys? Can anyone tell me? Streptococcus pneumoniae, right? Everyone know that. Streptococcus pneumoniae. Streptococcus pneumoniae. In that, there are the two strains are there. One is a S strain and another one is a R strain. Right, guys? So where S strain is the virulent one, whereas the R strain is the non-virulent one. Non-virulent one. Right. So what they have proved is, so they have injected both the strain into the mice. So when they have injected the virulent strain, so then mice was died. Right, guys? Why S strain is virulent? Can anyone tell me, guys? Why the S strain is virulent? Good morning, Varun Niramat, Shanta. Can anyone tell me why the S strain is virulent whereas the R strain is non-virulent? Because of polysaccharide coat, right? Because of polysaccharide coat. Polysaccharide coat, right? This is a S strain bacteria. This is the R strain bacteria. So where S strain is bacteria is having a polysaccharide coat on it, right? So that's why it is a virulent. Whenever we inject this into the mice is going to be die, right? So when he injected the R strain, the mice was alive because it is a non-virulent, right, guys? Because it is not having any type of the polysaccharide coat, right? So then when he injected the heat killed S strain along with the R strain, so here R, because the mice was died here, right? So then he came to know that what he did that heat killed, right? Heat killed S strain plus R strain he was taken. Now the mice was died, right, guys? So then it was heat killed. Already S strain, yeah, it is not having anything, right? Bacteria was died, but R strain is living. So then how come the R strain was converted into the S strain? So then they come to know that, so there are DNA is there in the S strain. So from this DNA, mRNA is going to be synthesized. So from this mRNA, protein is going to be synthesized. So that is helpful in forming the polysaccharide coat on the rRNA, right? Polysaccharide coat on the rRNA, then converting this non-virulent one to the virulent, right? Non-virulent one to the virulent one. So they have come to know that they didn't told that DNA is only the transforming here. Okay. So on that time, they don't know what is the DNA. So just they told that something is there which is transforming the R strain, non-virulent one into the S strain. That's it, guys. So that is the experiment was done in the 1928 by the Griffith, right? So this is the first experiment. So later, some experiment was done. Good morning, Varun. Some experiment was done in the year 1933. 1933, guys. After so many, after like 5 years, in the 1993 to 1944, the experiment was done by the MacLeod, McCarthy, and Oswald Avery. Oswald Avery. So these three of them conducted the experiment. So MacLeod, McCarthy, and Avery, Oswald Avery. Right. So what they have did, what is the experiment name they have did, guys? Year, always remember, in the multiple choice, they may ask you year, they may ask you the experimental name, or they may ask you the scientist name. Remember, guys. Okay. So the year was 1933 to 1944, MacLeod, McCarthy, and the Oswald Avery. So they have did the biochemical analysis of the transformation principle. Okay. So they have told that in the transformation principle, Griffith has told that something is going to be transferred. They don't know what is it, right? So here, MacLeod, McCarthy, and Avery they have confusion that whether DNA is transferring or RNA is transferring or protein is transferring. So what they have did, they have used the RNAs, they have used the protease, and they have used the DNAs, right, guys? So what they have did, so they have taken the heat killed. So they have did the biochemical proof. Just think that biochemical proof. Okay, fine. What they have did is they have taken the heat killed S strain. Okay, they have taken heat killed S strain, and for this they have added the protease. Okay, guys, they have added the protease. Then they have injected into the mice. Okay, the mice was died. Okay, the mice was died because the DNA is responsible for that, right? So the protein is going to be degraded, means nothing is going to be happen, right? So the mice is living only. Okay, sorry, the mice is living, not died. Mice is living. Okay, so then they have taken the heat killed estate. Then they have taken the RNAs. Then also mice is living. Then also there is a transformation of the R strain into the S strain is going to be possible. Then they have taken the DNAs. So then they have injected that into the mice. Then the mice was died. When they have used the DNA, then the mice was died. Okay. Then they have come to know that here the transforming molecule was not protein and RNA. The transforming molecule was DNA. Okay. So in the molecular basis. But what happened here in the biochemical analysis? But what happened here? They're unable to prove by the experiment. Okay, guys. So they're unable to prove by the experiment. So that's why this experiment was not considered as a proof. Okay. So then again one more experiment was done. In this experiment, they have proved that DNA is a genetic material. So that is in the year 1952. Okay. So they have taken radio labeled. Okay. So they have taken radio labeled phosphorus and the sulfur. Okay, always remember, guys. They have labeled the protein. They have labeled the protein with the sulfur. Okay, and they have labeled the DNA molecule, genetic material with the phosphate. Can you tell me which are the amino acid that is containing the sulfur, guys? Can you tell me which is the amino acid consisting of the sulfur? Sulfur containing amino acid. Answer me in the chat box, guys. Fast. Which is the sulfur containing amino acid? Methionine and the cysteine, right? Which is the amino acid which is helpful in the forming the double bond in the protein? Cysteine. Cysteine. Remember, guys, cysteine. Okay, fine. So, in the 1952, the scientist name Hershey and Chase. Hershey and Martha Chase. Hershey and Chase experiment, we will call it as. Okay. They have taken the radio labeled. So the DNA they have labeled with the radio labeled phosphate, and for the RNA they have labeled, sorry, for the protein they have labeled with the radio labeled sulfur. Okay. So then they have conducted the experiment by taking the E. coli and bacteriophage. E. coli and the bacteriophage. Here they have labeled the DNA of the bacteriophage with the phosphate and protein coat of the bacteriophage with the sulfur. So then they have let that bacteriophage to infect the E. coli bacteria. Then they have observed that, so this protein coat is not going inside the bacteria, but only the DNA is going. Means now you tell me inside which labeled we are going to get, either the labeled sulfur, or we are going to get the phosphate labeled. Can you tell me, guys, which radio label we are getting, either the phosphate or the sulfur? Phosphate, right? Because protein coat is eliminating, right? It is detaching. Only the DNA molecule is coming inside the bacteria, so that is containing the phosphate group. So then they have come to know that the genetic material is the DNA, right? The bacteriophage, so that is sending the DNA. So then they have come to know that the genetic material is here DNA. Okay, guys, fine. So that is in the year 1952. By this experiment, they have proved that DNA is genetic material. Remember, guys, very, very important. In the 1952, Hershey and Chase have proved that DNA as genetic material. Genetic material. In the multiple choice, they may ask you the another question. Okay. So that is semi-conservative mode of replication. Okay, guys. The semi-conservative mode of replication was given by the Michelson and Stahl. Remember, guys. So that is in the year 1958 by the scientist Michelson and Stahl. Okay, they have told that the DNA will be replicated by semi-conservative mode of mode of replication. Means what? So the parental DNA has to be replicated. So here the half of the parental DNA is going to be conserved, whereas the new DNA is going to be synthesized in the another strand. Whereas after the daughter cell, after the replication, so each daughter cell consisting of one parental strand and one new strand. So that is nothing but semi-conservative. Semi means half of the parental DNA is going to be conserved. Okay, guys. So that is what they have told in the 1958 by the Michelson and Stahl. Okay, guys. How many of you understood this experiment? How many of you understood this experiment? Give me a thumbs up if you understood me. People from the YouTube, give me a thumbs up or any answer in the chat box. So always remember, guys. So there are the 4 experiments there in search of the genetic information. So one is started in the 1928, that is a transforming principle by the Griffith. And second one in the 1933 to 1944, that is a biochemical experiment done for the transformation principle. They continued the transformation principle only. Okay. And in the 1952, they have told DNA is a genetic material in the 1952. Remember, guys. Okay. And in the 1958, Michelson and Stahl proved that DNA replicated by the semi-conservative mode of replication. Okay. Can you tell me who told that DNA is going to be? They have proved, right? Michelson and Stahl proved the semi-conservative mode of replication. Can you tell me in which year, which scientist has told that DNA replication is by semi-conservative? I'm asking you that who told. I'm not asking you that who proved. Remember, guys, proved is different, told is different. Can you tell me, guys, who is the scientist? You two people from the chat box, tell me, guys, who is the scientist? Who told the DNA replication is a semi-conservative? Nandish, try. Who's the scientist? Shanta, can you tell? You two people, answer me in the chat box. Fine. student in the exam is the as who is the scientist who discovered the replication of the DNA is nothing but our Watson and Crick. Okay, that is in the year 1953 when they have described the structure of DNA. On that time only they have told that they have observed that the DNA is replicating by the semi-conservative mode. Okay, but they have not proved that. But is in the 1958 Metzelson and Stahl proved that. Okay, proved is different and told is different. Always remember. Okay, fine. So let us go to the next. So the central dogma of molecular biology. Everyone know that. So it was told by the Frederick Griffith. In the multiple choice, they may ask you. Okay, remember guys. The central dogma of molecular biology was given by Frederick Griffith. Sorry. So that is Francis Crick. Okay, Francis Crick. So what they have told is nothing guys. So the DNA is present in the nucleus. The information which is present in the DNA, it is utilized for synthesis of the mRNA, right or not? mRNA. So then the information which is present in the mRNA, it is utilized for protein synthesis, right or not guys? So that is what we will call it as central dogma of the molecular biology. Other than that, there are the another things are there. So let us learn about that. If the DNA is going to be replicated, so that we will call it as a DNA replication. We require the DNA dependent DNA polymerase enzyme, right guys? So the DNA, the information present in the DNA is converted into the mRNA by the process of transcription. So here which enzyme is required guys? From the DNA, we are going to synthesize RNA, right? So then the enzyme should be DNA dependent and RNA is going to be synthesized. So that's why the polymerase should be RNA polymerase, right? So that's why the enzyme name is DNA dependent RNA polymerase. DNA dependent RNA polymerase in the transcription. Then the information which is present in the mRNA, it is utilized for the synthesis of protein. So that process we will call it as translation. Here we require an enzyme. Always remember guys, for the translation, always everyone remember that ribosome is required, ma'am. But the enzyme which is required for the translation is peptidyl transferase, or we will call it as the enzyme required is peptidyl transferase. Peptidyl transferase, and also we will call it as ribozyme. Ribozyme. It is RNA containing enzyme. RNA enzyme. Okay, guys, it is an RNA enzyme. Very good, Nandish. Yes. So then let us go to the replication. So what is replication, guys? Exam is nearer. I have not made any notes. Okay. So what I will do? Nandish has written very good notes. I will take the notes from the Nandish and I will make a copy. I will make a Xerox copy of that. Right. So then I can use it for the reading, right? Yes. But what happened in the replication? The same thing, guys. Okay. We have one parental DNA. Just we want to make a copy of that. Okay. Why we require a copy of that? To give it for the daughter DNA, right? To give it for the daughter cells, right? Yes. So how the replication is going to be taken place? So, and now you tell me guys, actually I don't write the notes, ma'am. Is it okay, Nandish? Then I will not take your notes, I will take the Shanta notes. Fine. So now you have to tell me guys, where the replication is going to be taken place in the prokaryotes. It is in the cytoplasma, right? Our prokaryotes, Papa, they don't have any types of the membrane bound organelles like the nucleus and all the things. So that's why the DNA replication in the cytoplasma. Then coming to the eukaryotes, so they are like a rich people, right? So they are having the membrane bound organelles. They have the separate compartment for the DNA synthesis only, right, guys? So the DNA synthesis will take place in the nucleus, in the eukaryote, right? So now we came to know that where the DNA replication is going to be happen. Now let us start the replication. Okay, guys. So now see here, DNA is there, right? DNA is very large. We know that DNA is very large, right? So see here. So where the replication is going to be taken place? From the random position? No, in the random position, the DNA replication will not take place. Okay. There are oocyte is there. Oocyte means what? Origin of the replication is there means. Okay. Here you can see this is the DNA. There is the origin of the replication is there. So from here, the replication is going to be started. Okay. So how the replication is going to be started? Which is the main enzyme, guys, in the DNA replication? Can anyone tell me? And one more thing you have to remember, the main enzyme is the DNA polymerase enzyme, right, guys? And origin of the replication, ori site will be a single in the prokaryotes. When you're talking about the eukaryote, there are so many origin of the replication sites are there in the eukaryotes. Remember, guys, there are the multi ori sites in the eukaryotes, whereas the single ori site in the prokaryotes. Okay, fine. You all know that the DNA is going to be replicated by the semi-conservative mode of the replication. It is given by the Messelson and Stahl in the year 1958. Okay, fine. So then what are the things we required for the DNA replication? Okay, let us see first. First, I will tell you how the DNA replication, what are the enzymes which are required for the DNA replication. First, we require the helicase enzyme, guys. What is the function of the helicase enzyme? So there are double-stranded DNA are there, right? Double-stranded DNA is there. So we have to break that hydrogen bond, right? Between the doubles, we have to make that into the single strand, right? For copying, we require the single strand. For example, this is a double strand DNA is there. What we have to do? We have to separate that, right? So how we will separate? We will separate like this, right, guys? So here, see here, I have separated, right? So for this separation, I require a helicase enzyme. Helicase enzyme, right, guys? So here you can see between these two polynucleotide chain, there is a hydrogen bond is there, right? The helicase, while unwinding, it will break hydrogen bond, right, guys? Can you tell me how many hydrogen bonds are between the adenine and thymine? Adenine and thymine consisting of how many hydrogen bonds? Yes, 2. Whereas the cytosine and guanine consisting of 3 hydrogen bonds. So these hydrogen bonds are cleaved by helicase enzyme, guys. Helicase enzyme. Okay. So when this hydrogen bond was cleaved, so now we have got the single strand DNA, right? Single strand DNA we have got now, right? So see here, then another enzyme. So that is the DNA gyrase. DNA gyrase enzyme that relieve the stress which is created by the unwinding. So then single-stranded binding protein, primase enzyme, DNA polymerase 1 and DNA polymerase 3 and the DNA ligase enzyme we require. Let us talk about the DNA replication. See here, guys, the DNA replication start in the particular site. We will call it as ori site. So for example, this is the ori site. So for the ori site, the DNA helicase started to unwind. Okay, started to unwind. See here, unwinding was started by this is the helicase enzyme. Okay, guys. So now see here, the single-stranded DNA was exposed, right? This is a single-stranded. This is a single-stranded. So what happened, guys? It will open DNA helicase. After some time, what will happen? The DNA will again wind up. Again, it will wind up. Okay. So that is the reason till the replication was completing, it should be the single-stranded only. For that, the single-stranded binding protein will come and bind. See here, guys. Single-stranded binding protein will come and bind to this opened single-stranded DNA. Okay. So now what will happen? So the primase enzyme will come. What is the first enzyme, guys? The first enzyme will be helicase, right? So after that, single-stranded binding protein will be come and bind to the single-stranded DNA, right? Single-stranded DNA, right, guys? See, observe here. So the junction between the two single-stranded DNA and the one double-stranded DNA, we will call it as what we will call it as, guys? Loop, replication loop. We'll call it as a loop. Okay. So, sorry, 1 minute. Replication fork, not loop. 1 minute, guys. Yeah. So, what we will call it as replication fork. Replication fork. This is what we will call it as a replication fork. Okay. So, after there is another enzyme will come. So, you know, our DNA polymerase 3 enzyme is very attitude. Okay. Very good, Lakan. Very good fun. Okay. Our DNA polymerase 3 enzyme is very attitude enzyme. Okay. It will not come, it will not start the DNA replication. So which is the enzyme is coming first? That is the RNA polymerase. Okay. RNA polymerase, Papa, it will come and first it will synthesize the it will first synthesize the RNA, guys. That RNA we will call it as primer. Primer is made up of RNA. Okay, guys. Primer is made up of the RNA. It will be of 5 to 7 nucleotides. Okay, guys. It will be about 5 to 7 nucleotides. So now our DNA polymerase will come and bind. So why our DNA polymerase is coming and binding here? Because it has got 3' OH, right? Without the 3' OH, our DNA polymerase cannot bind and cannot elongate. Okay, so that is the reason. So when the primer was synthesized by the RNA polymerase enzyme, so then the DNA polymerase has got the 3' OH. Then it is started to bind and started to synthesize the DNA. Okay, guys. Now it is elongating. So now you tell me, guys, is that replication is an energetic process or not? Whether the replication requires the energy or not? Can you tell me, guys? Yes. So helicase we will also call it as the unwind. Oh, sorry. This topoisomerase we will call it as the unwinding. Yes. Can you tell me, guys? Is that replication require ATP or not? Yes or no? Tell me in the chat box. YouTube, guys. We require energy. Where is the energy is coming? Where is the energy is coming, guys? Always remember. Always remember. So when the nucleotide is there, so we will call it as ATP. For example, adenine triphosphate, guys. Right? ATP. Right? How many phosphates are there in the ATP? How many phosphates are there? Three phosphate. Right, guys? 3 phosphates are there. Right? So when we added that, when the DNA polymerase add the nucleotide during the elongation, it is consisting of only the one nucleotide. Okay, it is consisting of only it is consisting of only one phosphate, where 2 terminal phosphate has going to be removed. Okay, so what happened in the 3 phosphate? 1 phosphate is going to be in the chain. Okay, whereas the 2 phosphate has been broken, right? Has been broken and removed. While brokening and removing, so these give the energy. Okay, guys. When this 2 terminal phosphate was removing, it will give the energy for the replication. Okay, guys. Always remember, in the 3 ATP, only 1 phosphate is going to be added in the chain, in the DNA polynucleotide chain, whereas 2 nucleotide was removed while adding the nucleotide by the DNA polymerase 3 enzyme. So that will give the energy for the replication. Okay, fine. So then how many nucleotide it can add, guys? How many nucleotide can add? I will ask you how many chapters you will read for the 1 hour. How many chapters you will read for the 1 hour, guys, or for the 1 day? You will tell that, ma'am, maximum three chapter you will read. Right, guys? During that exam time, so you will read more than 5 to 6 lessons, right? So here the DNA polymerase will add about 2000 nucleotide per second. Always remember, guys, 2000 nucleotide per second it will add. Okay, so how many nucleotides it is going to be add? 2000 nucleotide. 2000 nucleotide, that is per second, guys. Per second only it is going to be at the 2000 nucleotide. Okay. Now the elongation was started. It goes on elongating the DNA. So now there should be a termination should be there. Okay. Termination should be there. It is completed the DNA synthesis. Okay, guys. Now you tell me. So our double-stranded DNA is there. So that is having the RNA primer also. RNA. Right or not, guys? It is having the RNA also. Now we have to remove the RNA primer or not? We require to remove the RNA primer or not? Yes or no? We have to remove RNA primer, right? Then who is going to be removed? Our DNA polymerase 3 is going to be removed? No, guys. Our DNA polymerase is an attitude enzyme. It is not going to be come and remove that. Okay. So which is the enzyme is coming? Our DNA polymerase 1. Okay, remember. Our DNA polymerase 1 enzyme will come and it will remove the RNA primer. Okay. It will remove the RNA primer, guys. But who will synthesize the DNA in that position? Is that our DNA polymerase 3? No, guys, no. Our DNA polymerase 1 only will remove the primer and it will only add the DNA to that position. So now we have the complete double-stranded DNA, right or not? Tell me. Now we are having the complete double-stranded DNA, right? Yes. So finally we are having the complete double-stranded DNA, but there is a nick. There is a gap is present between the. So the RNA, that is the DNA polymerase 1 synthesize the DNA, whereas the DNA polymerase 3, for example, see here, it is removed, right? So now what will happen? So the DNA polymerase one also synthesize the DNA. There is a gap is there, right? So there is a gap is there. Who is going to be fill that gap? Who is that going to be join that gap? That is a DNA ligase enzyme. DNA ligase enzyme. Okay, guys. So that's it, guys. In the DNA replication, so there are 3 steps is there. 1 minute. There are the 3 steps is there. One is the initiation. Initiation. Initiation is going to be happen in the ORI site. The first initiator here is the helicase enzyme. Helicase enzyme will unwind, right? Unwind. So then we are going to, then the RNA polymerase is going to be synthesize the primer. So where the primer is going to be synthesized? At the 3' end. At the 3' end. So for example, if you take the DNA, so it is a 5', 3'. This is the 3', 5'. So near the 3', the RNA primer is going to be synthesized. Right, guys? Near the 3'. Okay. So if the near the 3' means this is the 5', right? So the DNA polymerase will synthesize the DNA in the 5' to 3' region. Right, guys? 5' to 3' region. Always remember, DNA polymerase always works in the 5' to 3' direction. Okay, fine. So this is we have studied. So these are the steps we have studied. First one is the recognition of the initiation codon. That is the ORI site. Then second one is the unwinding of the DNA. Then we will got the replication 4. Then the template DNA was separated. So now the RNA primer will bind and it will synthesize the DNA. Right, guys? Fine. So then it will elongate. Okay. So now here you have to remember always. So here the people will get the confusion in the which is the template for the lagging strand. So here you remember. So there is a 3' and 5'. 3' and 5' end template. It is act as the template for the leading strand. Leading strand. Remember, guys. Leading strand template is runs in the 3' to 5', whereas the lagging strand templates runs in the 5' to 3'. 5' to 3' is the lagging strand template. Lagging strand template. Right, guys? You all know that the DNA. So the leading strand, it is a continuous, right, guys? Leading strand synthesis was the continuous. It is towards the replication fork. Always remember, see here how the leading strand is going, guys. It is replication fork, right? It is going towards the replication fork, right? Always remember, leading strand will be always towards replication fork, towards replication fork. And coming to the lagging strand, we know that, so that is the discontinuously synthesizing. How it is going, guys? So it is a discontinuous synthesis, discontinuous strand, right, guys? So it is going away from the replication fork. Remember, so this is the replication fork. It is going like this, right? So that's why we will call it as away from replication fork. Remember, guys, away from the replication fork. Okay, fine. So Okazaki fragment. Okazaki fragment is going to be synthesized in the lagging strand, right, guys? We are telling as a lagging strand what happened here. So the RNA primer will synthesize. The DNA polymerase will synthesize some nucleotide and it will broken down. Means it will detach, not broken down, it will detach from the DNA. So then again primase will come and it will synthesize the primer. Then again the DNA polymerase will come. Then again it will detach. Then again primer, again DNA polymerase. It is like a lagging. Okay. So this thing we will call it as Okazaki fragment. Okay, guys. The Okazaki, now we have a question, guys. Okazaki fragment consisting of the only DNA or DNA and RNA? Tell me, guys. Okazaki fragment is consisting of only DNA or DNA and RNA? Fast. Tell me in the chat box. Lakan, tell me. Can anyone tell me? Always remember, Okazaki fragment consisting of both DNA and RNA. Right, guys? Why? Because RNA primer is there and small amount of the DNA was synthesized by the DNA polymerase enzyme. Right. So then it is consisting of both RNA and the DNA. Remember. Okay, guys. That's it. So how much RNA, how much Okazaki fragment will be there? So when we are talking about the prokaryote, 1 minute. Yeah. So when we are talking about the prokaryote, there will be a 2000, okay, 2000 Okazaki fragment will be there. Okazaki fragment. Okay, guys. In one breaking, there will be a 2000 Okazaki fragments are there. But when you are talking about the eukaryotes, it is having only 200 Okazaki fragment. 200 Okazaki fragment. Okay, guys. Fine. This is about the replication. Okay. So how many DNA polymerase enzymes are there, ma'am? So many of you will get confusion, right? So here is the answer for you. This is very important. DNA, what is the, there are three in the enzymes are there. One is DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. What is the function of DNA polymerase 1? It will remove RNA primer, right guys? It will remove RNA primer and it will synthesize the DNA nucleotide, and that is the main function of the polymerase 1. It is also having the proofreading activity, guys. But in the DNA replication, the proofreading activity is done by DNA polymerase 3 only. 3 only. Always remember, by the 3 only. Okay. Then DNA polymerase 2 is also there. So what is the function of DNA polymerase 2? You know mutation. You have studied in the inner strength biology, you have studied about the mutation. Right, guys? Point mutation, frame shift mutation, those are all the things. Right. So those are corrected by the DNA repair enzyme, that is the polymerase 2. Polymerase 2 we will call it the DNA repair enzyme. Okay, guys. Then polymerase 3, that is our main polymerase enzyme. Right, guys? Yes. Okay. That is all for this DNA replication. So here I have given some of the polymerase enzyme which are involved in the eukaryotes. That is not required for you. Okay. Fine. Then coming to the transcription. What do you mean by transcription, guys? All of you know that. In the central dogma, we have studied. So that information which is present in the DNA, which will synthesize it in the form of the RNA. Right, guys? So that is what we will call it as DNA transcription. Right? So it is a process of copying the genetic information from the one strand of the DNA. Always remember, guys. From the only one strand we are copying, not from the two strand. What happens if we copy from the two strand? Two mRNA will be obtained. If the two mRNA is obtained, means obviously it will be complementary, right? If the complementary means it will form the double bond. So if RNA only form the double bond means who is going to be synthesized the protein, guys? Protein is not going to be formed right from the double stranded RNA. That is the reason. And the another one reason is, so if the both the strands are coding means both the strand will code for the different protein, right? So it will create an anti-protein, different protein. That is also the one problem. Okay. So that is why we are taking only the one strand of DNA for RNA synthesizing. Okay, guys. You remember. Okay. Fine. So then let us talk about the how the transcription is going to be taken place. Always remember, guys, we know that DNA replication will be for the whole DNA. Whole genomic DNA is going to be replicated, right or not? But in the transcription, on the particular gene, for example, I will tell the gene which is required for the beta galactosidase. Okay, the gene which is required for the beta galactosidase, only that is going to be undergo the transcription. Okay, that is what we will call it as only segment of the DNA undergoing the transcription. Only segment, not whole DNA, only segment of the DNA. Okay, fine. So let us learn about that. Ma'am, what is the proofreading? Yes, Lakan, very good question. What is the proofreading mean? So our DNA is going to be synthesized the DNA, right, guys? See here, I will tell here. So Lakan, what will happen, you know? So this is a one strand of the DNA, this is another strand, right? The daughter strand is going to be synthesized in the 5' to 3' position. Here also what will happen? So this is a 3', 5' means, so it will also synthesize in the 5' to 3', right? So sometimes what will happen? Everyone will make a mistake, right? Our DNA polymerase will also in a hurry, what it will do some mistake. Instead of some nucleotide, what it will add some other nucleotide, it will add some mistake it will do. Okay, but it will be corrected by the same enzyme only. What it will do, it will come backward. Okay, so this is an enzyme, it will come backward, back stepping. Okay, it will come back stepping and it will see, did I made any mistake, it will come backward and it will see if there any mismatch was happened between the nucleotide. Okay, If it is nothing is there, then it will synthesize the other form. Okay. So in every form, what it will do, it will come back and it will see whether I did any mistake. Same thing you will do in the FST exam, right? Whenever the so many FST exams you are writing, in every FST exam, what you will come to know that whether I did any mistake. Yeah, what I did mistake here. I didn't read the correctly, right? Some mistake we will do during the reading, had to read twice, right? The same thing here also it will do. It will do the proofreading of itself. What is the work done by the DNA polymerase 3? It will do the proofreading. It will go backward and it will check whether all the things was correct or not. Okay, Lakan, that is what we will call it as proofreading by the DNA polymerase 3 enzyme. Do you understood, Lakan? Fine. So now let us go to the transcription, guys. I have told you that transcription is going to be done only in the segment of the DNA. For example, this is the DNA. Okay. So I want only this space. Okay. Only from this to this transcription has to be happen. Okay. From this to this DNA has to be transcribed and it should give the mRNA. Okay, guys. Only I need only this much. Only this much. Okay. So where this is starting, I will call it as initiator. Okay. Where it is ending, I will call it as termination, right? So in middle, what is there? My gene is there. Gene. Which gene I required? For example, LAC-Z gene I required. So that is a gene is present in the middle. Okay, guys. So that is what happened in the transcription. Only the segment of the DNA is going to be undergo the transcription. So how it is going to be happen? There's a double-stranded DNA is there, right? First, we will unwind that. Okay, first we will unwind that. See, observe here. There is an unwinding happen here, right? So see here, there is a double-stranded DNA. 1 minute. Observe here. So double-stranded is like this, right, guys? Helical structure. What I will do? I want only this much DNA should be transcribed, right? So what I will do? Just I will open that. Okay, guys. Just I will open that. Okay, guys. So that I will call it as transcription bubble. Okay, guys. Only this much of the DNA I want synthesis into the RNA. Okay. So this bubble I will call it as transcription bubble. Transcription bubble. Okay, guys. How it is going to be happen? Let us see. Okay. So here also there will be a 3-step is there. The same 3 steps. One is the initiation. Second one is the elongation, and third one is the termination. Okay, guys. What happened in the initiation? Let us see, guys. Okay. So what happened in the initiation? The main enzyme which is involved in the RNA synthesis, or the main enzyme which is involved in the transcription is, which is that enzyme, guys? Can anyone tell me which is the enzyme which is involved in the? That is RNA polymerase, right, guys? Whether it is the RNA dependent RNA polymerase or DNA dependent RNA polymerase. That is DNA dependent, because we are taking the information from the DNA, right? Then what we are going to synthesize? RNA. So that's why we have to call it as DNA dependent RNA polymerase. Okay, guys. So before going to tell what is the DNA, how the transcription is going to be taken place. So let us talk about the RNA polymerase, guys. Okay. RNA polymerase enzyme. So when you are talking about the eukaryotes, when you are talking about the prokaryote, there is a single RNA polymerase. It will synthesize all the type of the RNAs. How many types of the RNA are there, guys? rRNA is there, ribosomal RNA, tRNA is there, transfer RNA, and mRNA is there, right? Both of these 3 RNA in the prokaryotes is synthesized by the single RNA polymerase. Okay. So then how it is going to be function? So see here, it is going to be synthesized in the 5' to 3' direction only. Always remember, DNA replication is also in the 5' 3' direction only, and RNA synthesis also in the 5' 3' direction only, and protein synthesis also in the 5' 3' direction only. Okay, guys. Always remember, in the molecular biology, everything in the 5' 3' direction only. Okay, fine. So let us talk about the transcription unit. Transcription unit, I have told you, only this segment of the DNA I require for the synthesis. What are there in that segment? So observe here, guys. So there is a promoter. Okay, this is a segment of the DNA I require. So in that segment of the DNA, there is a promoter is there. Okay, guys. So one promoter is there, and so there is a structural gene. This we will call it as structural gene is there, and there is a terminator is there. Terminator. Okay, guys. So the synthesis will take place in the promoter, where RNA polymerase will come and bind. So there is the promoter region. Okay. So where in the exam they may ask you. So where is the RNA polymerase will come and bind? Is it going to come and bind on the DNA only? Yes, in the DNA only. Where? That is in the promoter region. Okay, guys. In the promoter region, it is come and bind, and it is going to be elongate. It is going to be synthesize the RNA. And where it is going to be terminate? For example, I started the class. Okay, I will start the class at the 10 o'clock. Okay. I will not stop my class. Should I continue the class over the 2 to 3 days continuously? Not possible, right? If I do the continuation of the class also, you will not listen, right? And it is not possible for me also to continue the class for continuous 3 to 4 days, right, guys? So where is the starting is there should be an ending is there, right? The same thing happen here. In the promoter side, it will start, whereas in the terminator side, it will stop. Okay. So in the terminator side, it will stop. So promoter side will be in the 5' region, and the terminator will always will be in the 3' region. Remember, guys, terminator will be in the 3' region. Okay. So when we are talking about the transcription, so there will be 2 strands of the DNA is there, right? Ma'am, from which strand of the DNA they are going to be copy the mRNA? I will tell you. So the one strand which is running in the 5' to 3'. Okay, guys, which is running in the 5' to 3', we will call it as non-template, and we are calling as coding strand. What do you mean by coding strand? mRNA, we have synthesized the mRNA, right? That mRNA is having same nucleotide as the coding strand. Okay, our mRNA will also run in the 5' to 3', whereas the coding strand will also having the 5' to 3', right, guys? So then, which is the template strand? So then, template strand has having the 3' to 5'. Okay, guys, which is the DNA is acting as a template, that is 3' to 5'. It is very easy to remember, guys. Okay, you know that. So the RNA synthesis will be happen in the 5' to 3'. Then obviously, who is the template? Tell me, guys, who is the template? Tell me, guys, in the chat box. So what is the template? What is the primes of the template? Tell me. The template will be running. I need answer from the YouTube people also. Answer me in the chat box, guys. Fast. Nandish, answer me. Lakan. How it is going to be? Obviously, 3' to 5' is a template, right, guys? Remember, don't confuse. 3' to 5' is a template strand. If it is a template strand, means it is a non-coding strand, right? It is a non-coding strand, right, guys? Right or not? We are taking the 3', 5' as a template strand. We are synthesizing our mRNA. So have you understood what is a transcription unit? Very good, Lakhan. Very good. Have you understood what is a transcription unit? Give me a thumbs up if you understood means. Yes, fine. So here the important, very important is this thing only, guys. So in the exam they may ask you what is the template for the RNA synthesis, that is 3' to 5'. Whether it is a coding or non-coding. Obviously it is non-coding strand. Okay. Coding strand should be same as the mRNA only. Remember. Okay. Fine. So let us come here. What is a UTR, guys? What is UTR? Untranslated region. Right, guys? UTR. UTR means what? Untranslated region. Okay, we will also call it as upstream UTR. Upstream UTR and the downstream UTR. Where is the upstream UTR going to be present in the exam? In some in one of the NEET exam, they were asked that the UTR is going to be present only at the 5' end. So that is after the promoter. After the promoter. Is that correct, guys? Is that UTR is going to be present after the promoter or before the promoter? Tell me. After the promoter or before the promoter? Tell me in the chat box. Always remember, guys, UTR present in both 5' and the 3' direction. Always remember. So UTR present in both 3' and 5'. Okay. So where it is going to be present? It is going to be present before promoter. It is going to be present before promoter and after terminator. After terminator. Okay. So why UTR region is required? It is required for the efficiency, efficient translation. Efficient translation. Remember, guys, it is very, very important. Okay. So where is the UTR is going to be present? See here. This is a one UTR. This is present before the promoter, right? In the 5' position. So here after the terminator, there is another UTR, another UTR. It is present after the 3'. So sorry, after the terminator. So UTR is helpful in the translation, efficient translation process. Okay, guys, fine. So let us talk about the how the transcription is going to be taken place. Always remember, guys. So however, DNA polymerase cannot start the process like that only. The RNA polymerase alone cannot synthesize the mRNA. Okay. So what is required here? So our RNA polymerase enzyme for the starting it requires another person, another friend, whereas the ending it requires another friend. Okay. For the starting it requires the friend, that is we call it as the sigma factor. For example, if you think the RNA polymerase cannot identify the where is the promoter. So that's why it will wear the chasma. Okay. So what is the chasma here? The chasma is nothing but the sigma factor. Okay, guys. So once it is find the promoter, it is like I don't want chasma now. Why I need chasma? It will remove the chasma. Okay, guys. It will remove the sigma factor after recognizing the promoter region. Okay, guys. So it will goes on synthesizing. Okay, guys. It is goes on synthesizing, and when it is reaches the termination. Okay, guys. So when it is reaches the termination, it unable to terminate. It is unable to know where I have to leave, where I have to leave the place. Okay. So it is was searching where I have to leave the place. It is unable to remove. It is unable to terminate the RNA synthesis. What it will do? It is require the another friend. What is that another friend, guys? That is nothing but we will call it as P factor, or we will also call it as rho factor. Rho factor. Okay, guys. For the starting, it requires the chasma to identify the promoter. The chasma name is sigma factor. Okay. So the RNA polymerase will bind to the sigma factor and synthesize only for finding the promoter, guys. So when it is elongating, it will remove the sigma factor. Observe here. So when it is going to be elongate, it removes the sigma factor. Chalo. I will not look nice when I am wearing the chasma. So now I don't want the chasma. It will remove. Okay. So after that, the termination side, it come to know that now I have to get down. I don't know how to get on. I don't have chasma also, but it is having the another enzyme. So another factor, that is the rho factor. Rho factor, which is helps in the termination, guys. Remember, okay, which is the enzyme which is involved in the transcription. From the DNA we are copying, right? So that's why it is DNA dependent. So we are synthesizing the RNA. So that's why RNA polymerase. RNA polymerase. Okay, guys. Fine. So this is what I have told you. So during the elongation, you know that. So in the RNA we will use the nucleotide. So that is the ATP, GTP, TTP, and the CTP. In the DNA, what we will use nucleotide? So that is the DGTP. So that is DGTP, right, guys? DCTP. Whereas DTTP, where in the DNA replication, always remember, in the DNA replication, we are using the deoxyguanine triphosphate, deoxycytosine triphosphate. Okay, guys. But in the RNA, we are using adenosine triphosphate, guanine triphosphate, thymine, and the cytosine. Okay, guys. Fine. So now I have told you that in the termination in the bacteria, there are two types. Okay. One can be by Rho factor and another, it does not require the Rho factor. What it will do? It will form the hairpin loop like structure. Hairpin loop, hairpin loop, hairpin loop. Okay, guys. What is a hairpin loop? See here. So, which is rich in the GC. Okay. Some of the strand was synthesized by the RNA polymerase enzyme. At the end, what it will do, where the more GC rich is there, so there it will form the bonding. Okay, it will form the loop-like structure. So then, so the RNA primers will form the heavy-like structure, right? So it will remove from the synthesis. So the RNA was removed from the RNA polymerase enzyme. mRNA was removed from the RNA polymerase enzyme. So that is detached. Okay, guys. So this is about the transcription. How many of you understood transcription? Tell me, guys. How many of you understood transcription in the chat box? You two people, give me a thumbs up if you understood, or tell me whether you understood or not. Where is the transcription is going to be taken place? Can anyone tell me, guys? Where is the transcription is going to be taken place? Either in the cytosol or in the nucleus? Either in the cytosol or in the nucleus? In the nucleus. Remember, guys, replication and also the transcription will take place in the nucleus, in the nucleoplasma, nucleus. Okay, fine. So then we have come to know that. So what do you mean by gene and what do you mean by cistron? Always remember, guys, what do you mean by gene? Gene is nothing but, so that is a functional unit of the inheritance which is present on the DNA. Okay. So it is a sequence of DNA. Either it may code for hnRNA or mRNA, or it may code for the rRNA, ribosomal RNA, or it can code for the tRNA. So that we will call it as a gene. Gene may code for anything. Okay. Either mRNA, tRNA, or the rRNA. But when we are talking about the cistron, it is a segment of the DNA which is code for polypeptide. Remember, guys, it is a segment of DNA. It will code for only for the protein, only for the polypeptide. Okay, guys. So that is what we will call it as a cistron. So gene may to it may synthesize any RNA, mRNA, tRNA, rRNA. But cistron will give rise to only the polypeptide. Polypeptide. It is a segment of the DNA which will synthesize polypeptide. Do you understood this? Yeah, Lakan, in bacteria cytosol only. In bacteria, we don't have nucleus, right? In bacteria, for example, if you take the prokaryote, it is going to be synthesized in the cytosol. DNA replication, RNA synthesis, transcription, everything in the cytosol. When we are talking about the eukaryotes, DNA replication is in the nucleus. Okay. And RNA synthesis, that is the transcription. Transcription is also in the nucleus. Okay. Whereas the protein synthesis, or we will call it as the translation. So that is in the cytosol. Okay, guys. Fine. Have you know what you mean by monocistronic and polycistronic DNA, guys? DNA? See here. So when we are talking about the monocistronic, mono means what? Single, right, guys? Single means what? See here. This is a gene. So this is a one promoter. This one promoter is having the only one gene. Okay, guys. One promoter is there, whereas the one gene is there and the one terminator is there. Okay. So those type of the genes we will call it as monocistronic genes. Okay. So we can see monocistronic genes only in the eukaryotes. In the eukaryotes, we can see the monocistronic DNA or monocistronic gene, not a DNA, gene. Okay, guys. What do you mean by polycistronic? Polycistronic. In the polycistronic, what will happen? See here, one gene, one promoter is there, guys. One promoter is there. But for one promoter, there are more than one gene is there. See here, one gene, second gene, third gene, and later the terminator is there. Okay, where one promoter is having more than 2 or 3 genes, we will call it as polycistronic. Polycistronic. Cistron means what? It is cistron means what, guys? It is coding for the polypeptide, right? See here, in the monocistronic, it is coding for the single peptide, right? So that is monocistronic. But what happening in the polycystonic? It is having the 1, 2, 3. Right, guys? So that's why we will call it the polycystonic. Polycystonic is going to be present only in the prokaryotes. Always remember, guys, prokaryotic mRNA is polycystonic, whereas the eukaryotic DNA is the monocystonic. Okay, guys? That is what they are given here. Okay, fine. In the monocystonic DNA, so there is the introns and exons are present, guys. Okay, what do you mean by introns and the exons? Introns, if you take, this is the monocystonic DNA, it is having the exons and the intron, guys. See here, whichever the thing I am making the blocking, so that are the coding DNA. Okay, these are only coding. So those coding we will call it as a exon. Remember, guys, coding DNA we will call it as exon. Other than that, in between, non-coding is there. Non-coding DNA part, that we will call it as the intron. Intron. Okay. In the monocystonic DNA, that is consisting of both exon and intron. Exons are coding, introns are non-coding. So where our matured mRNA consisting of only the exon, guys. So where introns will be removed by the splicing. Okay. So then coming to the polycystonic gene. Polycystonic gene does not contain introns. No introns are present. Always remember, no introns are present in the polycystonic DNA because that is a prokaryotic DNA, right? Does not contain any type of the introns. Introns are present in the mRNA, guys. mRNA. Okay. So in the polycystonic mRNA does not contain any type of the introns. Okay. In the prokaryote. Prokaryotes. How many of you understood this, guys? Give me a thumbs up or tell me in the chat box, do you understood or not? Shanta, Nandish, do you understood? Sharda? Fine. Always remember, guys. Mono means single, that is going to be present in the eukaryotes. Poly means it is coding for so many more than 2 to 3 more than 2 to 3 enzymes or the protein. So that is going to be present in the prokaryote. Fine, guys. So then coming to the transcription in the prokaryote. Simple, guys. So when the transcription is going to be happen, so before the mRNA should be completed, suddenly the ribosome will come and bind to the mRNA and it will synthesize the protein also. So that's why in the transcription in the prokaryote, we will call it as coupled process. Coupled process. How? Before the transcription is completing, the ribosome will start synthesizing the protein, right? So that's why because in the transcription is going to be taken place in the cytosol, there only the ribosome is going to be present. Where transcription and translation is coupled in the prokaryote. Coupled. Okay. Fine. Then coming to the transcription in the eukaryote, it is not coupled. Once the transcription was completed, so the processing should be happen, guys. Always remember, in the prokaryote, mRNA was came by the transcription, right, guys? But what happened in the, yes, lesson, I will complete by this thing. What happened in the eukaryote, the mRNA is not produced. The production of the transcription is only the hnRNA, heterogeneous mRNA, right? Heterogeneous nuclear RNA is going to be produced in the eukaryotes. What is that? We will call it as hnRNA, right, guys? We have got hnRNA, right? So this hnRNA is not the active, or we will call it that it is not the matured. It is not matured, right? What we have to do? We have to mature that. We know that this is having the exon and the introns in between, right, guys? Unwanted introns they are having. The introns are not going to be translated. Remember, guys. So that's why we'll remove that, right? So removing of the intron. Always remember, some of them will confuse between the exon and the intron. Intron is non-coding. Intron is non-coding. Okay. During the splicing, the introns are removed, guys. Okay, introns are removed, and they will add all the exon 1, exon 2, and the exon 3. They will add this all the exons. That is what we will call it as splicing. Some of them will think that splicing means cutting. No, splicing means joining the exons. Joining the exons, we will call it as splicing. Okay, guys. So now the splicing was happened. So now you can see here the cap. We are going to, in the 5 prime, we are going to be add the methionine. So that is the methionine guanine triphosphate at the 5 prime. That we will call it as the capping. Okay, guys. And at the 3 prime, we are going to add the tail. So that is the polyadenosine. So the polyadenosine will be a tail. So that will be of 200 to 3000. So that is by the enzyme PAP. PAP. What do you mean by PAP, guys? PAP enzyme. Always remember. The poly A tail is produced by the PAP enzyme. PAP means polyadenosine polymerase. Polyadenosine polymerase. PAP enzyme. Okay, guys. So tailing is done by the PAP enzyme. So that is tailing will be always in the 3' position. Then what is the essential for capping and also the adding the tail to the mRNA? So now our mRNA was matured. Now our matured mRNA consisting of cap at the 5' and tail at the 3', and it is not at all consisting any introns. It is consisting of only the exons, right, guys? It is consisting of only exons. Now we will call it as matured mRNA. Matured mRNA. Okay, guys. What is the useful of this? So it will first thing it will help in the transport of mRNA from the nucleus to the cytoplasma, right or not? It is helping in the transport of mRNA from the nucleus to the cytoplasma. That is the one thing. And so many enzymes are present in the cytoplasma. If the cap is not there, means it will cleave the mRNA. Okay, it will cleave the mRNA. So that is the reason we have to add the capping and tailing to inhibit the cleaving from the other enzyme. Okay, guys. So that's it about the transcription. If you are having any confusion, you can ask me. Are you clear, guys? Are you clear? Do you understood this all the thing? In the coming class, we will learn about that and all the ways for the tomorrow's mock test. Okay, guys? All the ways for the tomorrow's mock test. Do well. Okay, whenever you are giving the mock test, learn something from the mock test. Okay, guys? Do prepare. All the best. Thank you, Lakan. Bye. Do prepare well, guys. Your exam very near. After this chapter, I will do the human physiology. Okay? Fine, guys. Thank you so much. Take care.